Great. So, good evening, everyone, and thank you so much for joining in on this busy week. We welcome all of you here from Genova OncoCare for the Embrace Meet. So, before we begin the program today, just would like to give a small introduction of Genova. What are we as a company? So today we can say that in the last 10 years, we have made a difference in tens of thousands of humans of lives. But in the next coming time, we want to touch more than 1 million human lives as we progress ahead. So we aspire to transform the healthcare of millions of people, especially through implementation of technological solutions, and in process be the top 10 technology driven companies in the world. Currently, if we see, then we are a Pune based biotechnology company in the business of transforming healthcare. What we will do to do this first is manufacture biotherapeutics. Then, how we are going to do it is basically innovate biomanufacturing technologies. And why we want to do this is because we want to transform healthcare. So there are various milestones that we have achieved over the years. So Genova was established on 2nd of Feb 2006, and from there we have launched Elexim, MGRAS, 24, and over the years in 2024, the latest one was Hamsil Junior for pediatric cancers. So it's been a long journey for us. And we are having a lot of research going around the cancer care and management, mRNA vaccines and biotherapeutics, also in stroke management, etc. So with this, I welcome all of you to the role of Eribulin in management of MBC. We are going to, have wonderful moderators and speakers like Dr. Sham Agarwal. Sir definitely doesn't require any introduction from our end. Sir is the HOD of Medical Oncology at Sir Gangaram Hospital, Delhi. We welcome you here, sir, today. And the esteemed speaker, Dr. Ankit Jain, again a senior consultant, medical oncology at Apollo Center Care at Delhi. We welcome you here, sir, today for this wonderful session. With this, we are also having our expert panel of Dr. Mukesh Bansal from Haryana, Dr. Veenu Saharthi from Bangalore, Dr. Nikhil Shirsi from Pune, Dr. Nirmal Raut from Mumbai, Dr. Anupam Mahanta from Guwahati, Dr. Sanjeet Soni from Jaipur, Dr. Aditi from Rajkot, Dr. Mahanti from Guntur, and Dr. Durai Manavalan from Chennai. We welcome our esteemed panel as well. With this, I'll just take you brief through what the agenda for today will be. So for the first 10 minutes, we will have Dr. Kaivalya Kulkarni, our lead scientist at Genova, giving you a small perspective on Eribulin from the R&D perspective. So with this, I hand it over to Dr. Kaivalya. Over to you, sir.
Yeah, thank you, Dr. Ronak. I'll just would like to share my screen.
Yes, sir. Stop sharing from my end.
I hope the screen is visible to everyone.
Yes, the screen is visible, sir.
Yeah. Good evening, everyone. Welcome
Put it
to
on
this.
full screen mode, sir, before we begin.
Yeah. Do you see? I've already made it full screen.
It's not on the presenter mode yet, sir.
Do you see it now? Ronak, do you see it now? Ronak, do you see it now?
Sir? Not yet, sir. Can you try changing the slides?
One sec. Yeah, it's on the full screen at my system. Do you see screen changing or no?
Not yet, sir. Can you try resharing again, please?
Yeah, I'll possibly stop.
Apologies for the technical glitch, everyone.
Yeah, I'll reshare it.
Yeah.
Do you see it now?
Yes, sir. Please go ahead.
Okay, great. Uh, good evening, everyone. Welcome to the today's panel discussion. To start today's discussion, I would quickly like to give overview about erimulin mesylate uh, uh, development perspective. So we to start with, eribulin mesylate is a very complex drug molecule or API. It is known to be most complex API ever been synthesized in the lab by human beings. Although it's a derivative of a natural algae, this is the most complex drug ever synthesized. Uh, the, the complexity achieved by this drug is due to the presence of 19 chiral centers in the complex setup, and these 19 chiral centers would actually result into possible isomers which are more than half a million isomers. From this more than half million isomers, only one of the isomer is active uh, towards the indication, that is breast cancer. And so synthesis of this moiety in the lab or on the plant scale is huge task, or a very complex task, because it requires all asymmetric chiral center installations. This is all done using all natural carbohydrate precursors or shikimic acid pathway molecules. Uh, uh, the Embrema injection is actually made of is an intravenous solution with strength of 1 milligram per 2 ml. It is administered intravenously over 2 to 5 minutes on day 1, 8, and 21-day cycle. Let's move to our vision, how MQR has moved into eribulin space. So MQR or Genova launched Embrema as world's first generic in India. We are constantly developing our production to world-class quality standards for our domestic products. We are trying to match it to the innovator and all the world players. Right now, we are in process to apply this product in US FDA. We are already filed this product in Europe and Canada as well. Uh, right now, there are no US or European pharmacopoeia for this molecule. So we currently try to match our qualities, measure our API strength with the innovator product to ensure the quality standards. And we are trying to get this drug approved worldwide as well. So what we do to make sure that our drug product or API is best in class is that we use all top of the line pure techniques which are there, exist in the or available in the industry. One of that is the preparative purification technique. So this is basically a technology driven parameter. So therefore there is no chance for variation due to manual error or hand change. Good thing is that because there is it's all digitalized, everything is monitored online for the quality of the API which is made. And there is possibility or availability of iterative purification, which gives us the possibility of obtaining the utmost quality. Uh, and MQR has backward integrated the complete synthesis in our R&D and plant for production. Therefore, we basically make start to end to end synthesis of this API, and we have actually produced more than now 10 or 12 commercial batches with consistent quality. This has been continuous process, and we have been very successful about it. So in the last slide, I talked about the preparative purification technique. So what is it? It's a preparative HPLC, that is high pressure liquid chromatography. It is the technique which is used for higher quality. What happens is the it has a response towards the UV response towards the organic molecules. So when we see response UV response on this digital screen, we start seeing that our product is coming out of the column. So we start pulling the fractions, and then we check the quality, and the repeat purification cycles ensures equality. On the next slide, we'll see how this iterative purification technique works. So in the on the left hand side of the screen, we see a mobile phase. So mobile phase is pumped into the system through a pump. Then there is an injector, and then there is a column. So what happens is we actually inject our sample through injector, which is carried into the column using a mobile phase, that is organic and aqueous solvents. And once it passes through the column, like top we see in the first cycle, we see mixture of peaks which are coming out. And then what we do is we actually cut these peaks, collect these peaks into small fractions, and then we get a purer compound compared to our crude impure compound. And then it passes and gets collected in the flasks. Then what we do is we take back all of that solution, inject it again for the second time. And now in the second cycle, we see the center image that the peaks are separated slowly. So these peaks are collected again separately and collected. And what happens by the third or the fourth or the next rounds is that we get good separation, and we can easily desire cut out or separate the desired product peak from the unwanted peaks, and then start collecting. So and if for any reason, if this doesn't work in third or fourth cycle, we can continue this up to n number of cycles. The benefit of this is that we do not lose any material here. All the pure material is collected, all the impure material can be re-injected in the system, and we ensure that all the material is collected without being without losing any of the material. So that's how we make sure that the purification technique used gives us the quality which we require, and as well as we get the good yields without losing any material in the drain. Here is an example that when we start our crude sample, that is on the left, it shows that our desired peak is 46%. That's the first peak. And then our undesired peak is the second one, which is in total is about 53%. So when we carry out this iterative purification, slowly we start seeing that our small peak, which was there 46% in the first image, is now 99.36% pure. And all the integrated or when we add all the other peaks together, it's not more than 0.5%. So that shows the powerful technique or effect of the technique which we are using for purification of the API, that we reach from anywhere from 46 or 20 to more than 99.36 or more than 99.9% at times during purification. That gives us the edge over all the other players in the market. Now here is a quick review of data what we have done through commercial batches. The first data we see is the eribulin emblema from MQR, and then other next two are our API batches, and third is the RLD that we are comparing it with. So all throughout the first three batches, we see that the HPLC purity is above 99% compared to the RLD. It is better. Any unspecified impurity in the injection is 0.3% against APIs is not more than 0.3, or in second batch it's not even 0.1%, whereas it's in Halaven it's again 0.3%. And our total impurities are less than 1. And if we see the assays, our assays are consistently hitting more than 98, 99%, which shows that our specifications are as good as JP monograph specifications, and this complies our domestic as well as the regulated market specifications. With this, my part gets over. If anyone has any questions or queries, I'm open to answer. Thank you.
Thank you so much, Dr. Kaivalya, for this wonderful perspective from the R&D. And with this, you can stop sharing the screen. we move on to the next session of Dr. Ankit Jain, talking about the role of eribulin in metastatic breast cancer. Over to you, sir. Looking forward to your insightful talk.
Thank you, Genova team, for your kind invite for this meeting. And it's an honor to be here, especially with Dr. Shyam Agarwal chairing this meeting. So if you want to give me the control, I can put it on a slideshow, please. Yeah. So just before I start, let me give you a bit of background about myself. I'm consultant medical oncologist at Indraprastha Apollo Hospital. Now, after doing my graduation from MEMC in Delhi about 20 years ago, I moved to England and been there for the last 20 years, where I did my MRCP medicine, medical oncology, MSc clinical oncology, and was working there for the last 10 years as a consultant medical oncologist, and only moved to the country here about 1.5 years back. So today I'm going to be speaking to you about the role of eribulin in the treatment of metastatic breast cancer. So most of this has already been told to you in the previous presentation, but if I can speak a bit about eribulin. Now, this is a non-taxane, structurally simplified microcyclic ketone, analog of a natural product called halicondrin B, now, which was initially discovered in a marine sponge. Now, the way it works is by binding to the growing end of the microtubules, which leads to blocking their extension and prevents them from capturing the chromosomes and halting the cell division. Now, various mechanisms exist about how it works, as you can see from this cartoon, where eribulin also works in the tumor microenvironment, and it improves the microenvironment, making it from the suppressive to a responsive environment. And therefore, you can argue that it really helps the immune modulators or PD-1 inhibitors to work. And sometimes there are some data to show that eribulin can work with immunotherapy. Eribulin also reverses the epithelial mesenchymal transition, and this makes the cancer cells less invasive and more responsive to the treatment. Now, another mechanism, how it works is by causing the vascular remodeling and eliminating the tissue hypoxia, which improves the oxygen delivery and helps in the drug delivery as well, and which basically helps to better the tumor control and enhances the effect of subsequent therapy. So that particularly again shows that earlier we give this drug, it might help for other drugs in the future to work much better as well. So eribulin suppresses the new metastases in the metastatic breast cancer patients, and there was a study which was done to investigate this progression type of metastases in the breast cancer. And this was a retrospective study where about 66 consecutive patients were studied with metastatic breast cancer who had eribulin chemotherapy. And in all comers, it was seen that treatment before eribulin, about 49% of the patients progressed to metastases, whereas in eribulin treated with 25%, and this was a significant difference, as you can see from the p-value. And specifically looking at the luminal and the triple negative breast cancers, again, the similar kind of benefits, more in the triple negative, where the treatment from 75% progression before eribulin went down to 29.4 when the eribulin was given. And in the luminal, 43.8 versus 21.6, clearly showing that if you give eribulin, you basically delay the progression of development of new metastases, and which is clinically beneficial as well as beneficial in terms of improving the survival, as we will see from this pivotal trial called EMBRACE trial, which was a phase 3 open label randomized study, international trial. It was actually the first major single agent study of any cytotoxic or biological agent, which has shown increased survival in patients with patients who've been heavily pretreated for metastatic breast cancer. Now, as I said, it was a phase 3 study for locally recurrent or metastatic breast cancer, and randomized allocation 2:1, favoring the treatment arm, which is eribulin, given at dose of 1.4 milligram per meter square intravenously, is given on day 1 and day 8, 3-weekly basis. And it was compared versus treatment of physician choice, which about 96% of the patients had either gemcitabine, vinorelbine, or capecitabine. Now, if you look at the Kaplan-Meier curve, it really favored the eribulin with the median overall survival, as you can see, of 13.1 months versus 10.6 months, and a hazard ratio significant of 0.81. About 16% of these patients were HER2 positive, and about 19% were triple negative. So there was a fair distribution of different subsets of the breast cancer patients. Now, median duration of eribulin treatment was 3.9 months, and the median duration of treatment of physician choice when they were given chemotherapy was 2.1 months and 1 month when they were given hormone therapy. So clearly there was a significant difference in improving the overall survival and reduction of about 19% of the risk of death because of breast cancer when eribulin was given. The side effects, the serious adverse events occurred in about 25% in eribulin as compared to 26% in the normal control arm. So this was basically more or less comparable. The adverse events leading to discontinuation was 13% in eribulin, slightly less as compared to 15% in the control arm. And the grade 3 or 4 adverse events, which basically led to most of the side effects with eribulin, were neutropenia, leukopenia, and peripheral neuropathy as compared to the control arm. The progression-free survival again was significant, 3.7 months in eribulin arm as compared to 2.2, and hazard ratio of 0.87, which again shows 13% reduction in the risk of progression when patients were given eribulin. And if you see, there were 1% patients having complete response with eribulin, and most of the patients had either a partial response or stable disease, and less number of patients had progressive disease. And more patients responded with 12% objective responses in the eribulin arm as compared to 5%. And there was a clinical benefit rate also favoring the eribulin, about 23% as compared to 17%. So this is a drug which was working in improving the overall survival. It was improving the progression-free survival. More patients were responding. There were some complete responses. Most of the patients had partial or stable disease, and there was a clear clinical benefit as well. Subsequently, Professor Chris Twelves, who I had a pleasure to work with in the UK, did a pooled analysis of about 1,000 patients receiving eribulin as compared to 802 control. And as you can see, the hazard ratio in all subtype of cancer patients favored the eribulin. Overall population hazard ratio was 0.85. In HER2 negative, 0.82, similar in HER2 positive, and then in triple negative, 0.74. So patients, whether they had HER2 positivity, HER2 negativity, or triple negativity, they all were getting benefit from eribulin, whether they had visceral or non-visceral metastases, or whether they were taxane refractory or not, the benefit was clearly there. Though non-taxane refractory had a better hazard ratio, the non-visceral metastases had better hazard ratio as compared to visceral, and more than 2 organ involvement had better as compared to less than 2, but the benefit was seen in all the subgroups. The median overall survival in the pooled analysis was better than what we saw in the EMBRACE trial, 15.2 months as compared to 12.8, and the hazard ratio of 0.85. Subsequently, there have been some real-world data. Now, this is Mariam Jaffrey. Actually, my name is there on this one as well, because there were 14 hospitals across the UK when we took a real-world data and of patients who had eribulin for metastatic breast cancer. About 577 patients were included. The median age was 56 years, and there were about 73% of the patients having ER positivity. The median overall survival was slightly less than what we saw in the EMBRACE trial. It was 288 days. But when we look at the older patients, it was much more higher. In patients who had age more than 65 and above, it was 325 days versus 285 for patients who were younger at less than 65. The progression-free survival was 117 days. The key thing was that the median overall survival was much higher in patients who received eribulin much earlier, after fewer prior lines of chemotherapy, less than or equal to 2, with 328 days as compared to 264. So in this real world data, we basically showed that the patients had eribulin treatment being more effective in the earlier lines, and patients who were more elderly, they benefited more, and therefore these patients can actually be given eribulin without much of worry. So it kind of establishes eribulin as a choice of second-line treatment for metastatic breast cancer patients. Now, there was a TETRAS trial, which basically looked at the second-line eribulin in triple-negative breast cancer patients. Now, this was a retrospective multi-center study where 44 patients with triple-negative breast cancer were studied from 2013 to 2019, and they had eribulin in second-line settings across 14 centers in Italy. And as you can see, the 12 months progression-free survival was 17%. And when it comes to overall survival, 12 months overall survival was 43% and 24%, 13%. At 24 months, it was 13%. So it basically shows that early inclusion of eribulin in the continuum of treatment strategies could increase the inherent clinical benefits which we can achieve with eribulin. Then our own Indian study, which was done by Dr. Bajpai, it basically was a retrospective study of patients who was previously treated with metastatic breast cancer settings with at least 2 lines of prior therapy and received eribulin between 2013 to 2019. There was 78 patients included. The median number of previous chemotherapy lines here was 5, with a range from 2 to 7. So these were heavily pre-treated patients. The median PFS was 3 months, and the median overall survival of 7 months, basically showing that eribulin activity was unrelated to the number of previous lines and the type of metastatic involvement. And it was well tolerated, and only 2 patients had to discontinue treatment because of the severe toxicities in this real-world settings. So what are the latest advances? Are there some more data which is available in 2025 showing us about the use of eribulin? Now, there is a publication which shows the long-term outcomes of eribulin-based neoadjuvant chemotherapy for triple-negative breast cancer. This was a JBCRG22 study where patients were grouped based on their HRD status, which is a genetic marker, as you might know, which predicts the better response to treatment, and the germline BRCA mutated, another marker for the treatment response. So there were 2 groups. The first group A, which had HRD positive or BRCA mutated patients, and they were divided into 2 treatment arms. The first arm A1, which had paclitaxel and carbo, followed by anthracyclines, and A2, which had eribulin with carbo, again followed by anthracyclines. The group B, which were the HRD negative or more than 65 years of age, the 2 groups, B1, which had eribulin given with cyclophosphamide, and B2, which was eribulin given with capecitabine. And if the first 4 cycles of these did not work, then they were allowed to switch to anthracyclines. Now, 99 patients were followed up for about 5.6 years. And patients who developed pathological complete responses on the treatment arm, they had excellent outcomes. About 95% of the patients were disease-free, and about 100% of patients were alive after 5 years. And the patients who had non-pathological complete responses, they had some lower outcomes, with 71.4% having disease-free and 80.2% survived for 5 years. The better outcomes were seen when patients had high lymphocyte count or low neutrophil to lymphocyte ratio. Now, this is becoming very topical because we are trying to find out now whether low NLR ratio is much better prognostic indicator of the responses. And this is what we have seen even in the settings of immunotherapy. And it was clearly shown that this was the case even with eribulin. So eribulin-based therapy was effective, especially for patients who achieved pathological complete responses. And patients who had HRD positive tumors, they had over 90% survival after 5 years. Then there was another study published in June 2025, which was a multi-center observational study of patients with metastatic breast cancer treated with eribulin in 2nd, 3rd, or 4th line between September 2011 and June 2018. About 84 patients were enrolled. The responses were similar, irrespective of whatever settings of eribulin they were given treatment in, whether 2nd, 3rd, or 4th line. The median PFS was robust, about 9 months. The subgroup analysis showed that the similar kind of progression-free survival was seen irrespective of whatever line of treatment they were given eribulin in. And again, a robust median overall survival of 24 months. The main prognostic factors of overall survival was the duration of response and the positivity of the progesterone receptor, which was associated with better progression-free survival. Then again, last month, there was an abstract, which was a real world data with eribulin in HER2-negative breast cancer. And it was from the 2 centers in Spain, where about 211 patients were identified between the January 2016 and December 2022. These are the patients who had HER2-negative metastatic breast cancer treated with eribulin, and they were followed up until March 2024. And the median age was 62 years. About 2/3 of these patients had luminal phenotype. About 50% of these patients had high Ki-67 of more than 30%. And 18.3% patients were de novo metastatic settings. Now, based on the lines of eribulin, the patients had 6.2% receiving eribulin in first line, 17% second, 27% in third line, and 49% in fourth line or later. So there was a fair distribution of different lines of treatment patients received eribulin in. Overall, the progression-free survival was 4.76 months, and overall survival of 10.75 months, with a median follow-up of 12 months. And when we specifically look at the subtypes, the luminal phenotype, the PFS was 5.53 months and 3 months in the triple negative. So slightly better in the luminal as compared to the triple negative. And overall survival again was better in luminal, 11.43 as compared to 9.3 months in the triple negative arm. But both of these were significant, so clearly there was benefit more in the luminal than the triple negative arm. The overall was 43%, and the median survival was 7.6 months. The toxicities mostly were asthenia, neutropenia, and nausea. So overall, this basically showed that 211 patients, that the results were quite comparable to what was seen in the published data from the trials. And it basically establishes the efficacy and safety of eribulin in different lines of treatment of metastatic breast cancer patients, especially when they were HER2 negative. So what do guidelines suggest? Now, when you look at the NCCN guidelines, now depending on the patients, if they are HER2 positive and hormone receptor positive and HER2 negative, and if they have visceral crisis or endocrine refractory, in the first line settings, if they are BRCA positive, then you can give PARP inhibitors, but they are BRCA negative, or if they are HER2 IHC 0+, 1+, or 2+, so if they are basically HER2 low, then either they give systemic chemotherapy, or you can give trastuzumab deruxtecan. In the second line, if they are again HER2 low, then if you haven't given trastuzumab deruxtecan, then they can be given in the second line settings. But if they are not candidate for that treatment, then either you can use sacituzumab govitecan if you have excess or patients can afford. But if it comes to systemic therapy after anthracyclines and taxanes, then eribulin has a very well-established role, sometimes even ahead of drugs like capecitabine. And in the ISMO guidelines suggest in the patients with metastatic triple-negative breast cancer, if they have recurrence less than or equal to 6 to 12 months after the end of neoadjuvant immunotherapy or second and further lines. If they are BRCA mutated, then you can give PARP inhibitors or chemotherapy. And if they are BRCA wild type, then you can give sacituzumab govitecan or chemo. And on progression, if they haven't used sacituzumab, then you can use that. Or if they are HER2 low, then you can use trastuzumab deruxtecan. But otherwise, if it comes to chemotherapy options, then the choices are eribulin, capecitabine, and vinorelbine, with eribulin being a drug which can easily be used much ahead of capecitabine and vinorelbine based on the data which I've already shown you, and as well as their efficacy and the safety profile. So I think overall, there is now need to establish eribulin in a bit more ahead settings, and because of the efficacy which we have seen and the side effect profile. So I think with that, I finish my, can you see that?
Yes, sir.
Yeah. So with that, I finish my presentation. Thank you.
Thank you so much, sir, for such an elaborative and insightful session on the role of eribulin, especially in the setting of metastatic breast cancer. With this, I hand it over to Dr. Sam Agarwal to moderate the panel discussion. I will just load the slides at my end. Just a second.
So, thank you, Dr. Ankit, for that elaborate presentation on the use or the role of eribulin mesylate in patients with metastatic breast cancer. So it certainly adds a new dimension and a new agent in the armamentarium of oncologists for treating patients with the metastatic disease. So I believe there are some questions, so we need to interact with the elite panel which we have. I think they've been introduced. So I think let's go to the first question so as to, benefit from the discussion. So the question to Dr. Mukesh Bansal is that what percentage of your metastatic breast cancer patients are pre-treated in anthracycline and in taxane in neoadjuvant and adjuvant setting? So, or do you get patients de novo in the metastatic setting? Dr. Mukesh, what do you think? And then anybody can pitch in, please.
Sir, around 60 to 70% of my patients are pre-treated with anthracyclines or taxanes or with any form of chemotherapy. De novo metastatic breast cancer nowadays is reducing, and I don't think more than 1/3 of my patients are de novo metastatic breast cancer.
So the incidence of patients, coming up front with metastatic disease is declining. So anybody else wants to? Dr. Nirmal Roth is there. Would you like to comment, Dr. Nirmal, on this question?
So most of the metastatic breast cancers are ER/PR positive, so they would receive hormonal therapy first.
Right. But then, do you see patients who come to you with the metastatic disease at the outset?
Oh, yes, we
For
see.
example, triple negative patients,
Yeah,
who.
sometimes. But
So,
the
right.
incidence is less. More is of ER/PR positive.
So more is ER/PR positive.
Yeah.
And of course, these patients would be treated with the hormonal therapy first rather than
Yeah,
anthracyclines and with taxanes today in the era of CDK4/6 inhibitors. Anybody else? Dr. Vinu?
Yes, sir. Good evening, sir. So likewise, I think 70 to 80% we do see as post anthracycline and taxane treated, maximum 30%.
So I think we are getting a similar kind of a figure that majority of the patients would be sort of exposed to anthracyclines and taxanes before they, come to us for subsequent treatment. So can we have the next question, please? Okay, again to Dr. Vinu. In your clinical practice, where would you place eribulin for the treatment of metastatic triple negative breast cancer? I think we had a wonderful exposition from the speaker. So what is your current practice, in patients with TNBC?
Yes, sir. Currently, it's more likely post anthracycline and taxane failure, sir. But we do use it in second line in some of these patients who do present with de novo and have used taxane and platinum first. And then we quickly try to put them on eribulin as second line and try to extract maximum benefit there, sir. So mostly it's post anthracycline taxane failure, but also second line in people who present de novo, sir.
Right. Dr. Satyajit, I can see you on the screen.
Yes, sir.
Your comments, please.
Yes. Sir, same as Dr. Vinu, we are using as a earlier we are using like as a third line as in post anthracycline and taxanes, and now in some patients we also are using the second line.
So Dr. Mukesh, do you agree, or yours? What's the place of platinum, in patients with triple negative breast cancer?
Sir, yes, I agree. We use eribulin mostly third line and beyond. Also depends on the BRCA and PDL1 status. Coming on platinum, in the curative setting, we are adding platinum, but in metastatic, we are not very keen on adding platinum. We first go to our taxanes or anthracyclines, and then maybe depending on the patient performance status and disease load, maybe eribulin, gemcitabine, or platinum also sometimes.
So, Dr. Nirmal,
Yeah,
let me
yes.
give you a scenario that you have a patient of triple negative breast cancer who comes to you de novo, has not been exposed to any chemotherapy before. And the patient has visceral disease in the lungs and liver, but performance status is good. So what is the approach in such a patient? How do you start? And patient does have some family history of breast cancer.
I would send a HRR test to check for BRCA mutations first, and then I would start with Paclitaxel, cisplatin.
Okay, so Pacli
Cisplatin.
and platinum is going to be your first line.
Yes.
Anybody has a different,
That also on
thought?
weekly basis. Weekly would be better.
So you want to use a combination of two drugs,
Yes.
platinum and Pacli.
Taxanes.
So
Yes.
Mukesh, you agree? Dr. Mukesh, you
Yes,
agree?
sir. Yes, sir. I think if the patient is BRCA positive, then obviously platinum should be added.
Yeah, but the BRCA is going to take 2-3 weeks to come. So before you start, you know the BRCA, you have to start the patient on chemo.
I will start with taxanes as a single agent.
Right. So that is one question which could be, debated. And the other issue is, since this is a triple negative de novo, what's the role of CPS, the immune checkpoint inhibitors in these patients? So
Very-
some-
Yes, sir. We have to send the CPS if the patient has financial affordability. And if PDL1 more than 10, then obviously we can always go to immune checkpoint inhibitors.
Right. So Dr. Nirmal, do you agree with the immune checkpoint inhibition?
Correct. Yeah, we would add immune checkpoint inhibitions also.
So anybody else, commenting on first line, second line use of taxane, combination and a single drug? Dr. Anupam, I can see on the screen. Dr. Anupam Mohanta.
Yes, good evening, sir.
Yes,
Sir.
doctor.
Am I audible, sir?
Yeah,
Yeah.
we can hear you. Yeah.
Eribulin in the metastatic TNBC, first age will have to be considered. Usually we see it in a young patient. So the data is convincing to use it upfront, but in my clinical practice, I mostly use it second line or beyond. Those who are presented in de novo metastasis, or in metastatic disease, already most of the patients receive prior anthracycline and taxanes. In this case, I have used one patient eribulin for around 8 months with good results.
Right. Absolutely. So, coming back to that case, which is getting pembrolizumab and taxane. So, 9 or
Yes, sir.
10 months down the line,
Yes,
the
sir.
patient fails and there is a relapse. So and this patient is BRCA1/2 positive. So the question would be, so what would you choose between a PARP inhibitor, eribulin, or you still have not used anthracycline and platinum? So Nirmal, what do you think, in the second line setting after the failure of a taxane and immune checkpoint inhibitor?
I would not start this patient on only a taxane. I would always add a platinum in this, even if she is BRCA positive or negative, because we know that triple negative platinums do work. So I would use a platinum. And in case I'm not use platinum, I will definitely use platinum as second line.
Okay, so you want to use platinum
Yes.
in the second line after taxane if it has not been used. Dr. Satyajit?
Yes, sir. I do usually what do if I see usually disease burden is too extensive, and then I usually add platinum upfront with taxanes.
Okay. So I think there are
After seeing
people
all
who
the
are.
PD-1 and etc.
Basically, sir, based on the patient performance status and visceral crisis, if is there any visceral crisis is there, I would like to use dual agent, either paclitaxel carboplatin, to get immediate early response. if patient is stable and less disease burden, in that case, single agent is preferably like a taxane is my, usually we use taxane as a single agent. And strong family history, early age, strong family history, TNBC, likely there must be germline mutation or HRD positive. In that scenario also, I would like to prefer use of platinum. So it's based on case to case, visceral crisis, young age, triple negative, preferably platinum, I will add in the upfront setting.
Right. So in a patient who's BRCA2 positive, so after the failure of platinum and paclitaxel, so would you use eribulin or would you use a PARP inhibitor?
So PARP inhibitor will be my first choice because we know that the response rate will be higher, definitely.
Right. So overall, you would like to use a PARP inhibitor because of the BRCA2
Correct,
positive, ahead of eribulin. So eribulin will come in the third spot, right?
There are few new studies which have shown that eribulin before PARP inhibitor has increased the response of PARP inhibitor in patients with BRCA who are carrying BRCA mutations. But still, these are phase 2 and early studies, and still recommendation is those with carrying this mutation preferably use this PARP inhibitor before eribulin.
Okay. Anybody else? I think we haven't heard from Dr. Nikhil.
Yeah, I'm
or,
Nikhil only talking.
You are Nikhil. I can't see you on the screen.
Acha,
I
okay.
think Dr. Mahathi is with us. Yeah. Okay. Now I can see you. Sure. Or Dr. Aditi Thanki from Rajkot. Is she there? Okay, so I think we've covered the issue of sequencing of drugs to some extent. I think physicians would like to use immune checkpoint inhibitors if the CPS is more than 10 in conjunction with a taxane. Usually, there's a difficulty in affording immune checkpoint inhibitors, although they become relatively less expensive. Then the choice is to use single agent paclitaxel versus a combination of platinum and paclitaxel. I think that is possibly dictated by the disease burden and the symptomatology of the patient. And I think more than 80% of the patients will be BRCA1/2 negative. And I think eribulin comes in there in the second line after the failure of taxane. Although platinum is also to be considered in the second line if it has not been used in the first line. So I think that is where we stand. And then, of course, we do have other agents to follow in the subsequent lines. Next slide, please. This question has been answered. Anybody wants to take this, Dr. Nikhil? What about anthracyclines? Because in a de novo patient who has not been exposed to anthra, so where do you place anthra? Before eribulin or after eribulin?
Basically, anthracycline I can use in before eribulin also, but nowadays I'm using eribulin as a second line because anthracyclines are associated with high toxicity. Patient with PS is poor or including visceral crisis. In such scenario, I prefer eribulin first before anthracyclines. It's
Right.
case
Yeah, I think as we heard from the speaker, that the earlier line of use of eribulin does confer an edge in terms of PFS and overall survival. And I think one important aspect of eribulin is the ease of administration.
Correct.
Because it's virtually, does anybody use it in the OPD or in the daycare, Dr. Dirmal?
Yeah, we use it in the daycare.
Right. I mean, did you need a chemo port or a central line for using eribulin, or it can be given, as an intravenous bolus push in 2 to 5 minutes?
It can be given intravenous also.
Yeah. So yeah, I think the ease of administration of eribulin and maybe the lack of cardiac toxicity, very little nausea, vomiting, you know. So it's, I think of course, what about alopecia? Dr. Mukesh, you see alopecia?
Not significant, sir. Not a mild amount, yes, but not like anthracyclines.
Nikhil?
Sir, basically, definitely alopecia is less, but mostly we are using in the third line or second line where the alopecia is not the main concern. Here we are fighting with the survival. I think it is not the main concern, but definitely it's less compared to anthracyclines and paclitaxels.
Dr. Anupam, in your patient, did you get alopecia with eribulin?
Alopecia, sir, not much as compared to anthracycline and taxane.
Yeah, right. So I think we do see fair alopecia with eribulin, and I think especially in patients with triple negative, one has an edge with eribulin than as compared to anthracyclines. So one would, I think there would be a consensus by and large to use eribulin ahead of anthracyclines and capecitabine and gemcitabine and even vinorelbine, because these are the drugs, which are, so we've got a lot of sequencing to do. So depending upon the patient's performance status, so one can use these agents in a sequence. Next slide, please. Okay, so since we are talking about triple negative breast cancer, so what's your opinion on combining eribulin with the immune checkpoint inhibitor like pembrolizumab? So any thoughts, Dr. Nirmal?
Yeah, so personally, I've never combined eribulin with pembrolizumab, but given the CPA score of more than 10, I would want to combine.
So anybody has experience, Dr. Mukesh or Dr. Satyajit?
No, sir, not used till date this combination.
Yeah, I think there was a study conducted with a combination of eribulin and pembro. I think it was Keynote 150. It was a small study, and there wasn't much in terms of the hazard ratio favoring the combination. But I think it's a small study which needs to be sort of looked at large numbers. So I think you have to stick to the protocol of taxane with pembrolizumab in the first line for CPS greater than 1 in patients with triple negative breast cancer. Next slide, please. Yeah, so coming to we've discussed a little bit of toxicity. The issue is of peripheral neuropathy. And, what's your say, Dr. Anupam, because these patients, most of them would be exposed to taxanes, and even if somebody, has had neoadjuvant, platinum, so they would have peripheral neuropathy. So how toxic is eribulin on the peripheral nerves? Dr. Anupam, please.
Yes, sir. As far as grade is concerned, in my experience, both eribulin and taxane can have a similar grade. But as far as higher grade is concerned, taxanes, particularly paclitaxel, conventional, they do cause more neurotoxicity than the eribulin, sir. And importantly, eribulin I have been able to use particularly on those geriatric patients more than 65 years old, because those patients already have a pre-existing diabetes or other illness or previous taxane-induced neurotoxicity there. So in this scenario, this toxicity
Right,
is
so I think,
milder.
yeah. So there's going to be an issue in a diabetic with some, nerve damage and having been
Yes, sir.
exposed to a taxane. So, Nikhil, any comments or any patient where you had to reduce the dose or you could not continue eribulin because of neuropathy?
Regarding neuropathy, no, sir. I never reduced dose of eribulin concerning neuropathy. Definitely for neutropenia, I have to reduce or prolong the duration, but not for the as for neurotoxicity concern. Compared to taxanes, is much better and much less toxic.
Yeah, I tend to agree because eribulin post-taxane, it doesn't really worsen the neuropathy in a very bad way. Anybody else? Dr. Mukesh or Dr. Satyajit, please.
The eribulin has lesser peripheral neuropathy than taxanes, and even quality of life, I will say it's usually better with eribulin, especially in the diabetic patient or with pre-existing neuropathy.
Well,
Right.
my view is that we usually use eribulin in the later line. So it is very difficult to, or maximum patient, those on taxanes have already developed the neuropathy. Grade 1, grade 2 already they developed the neuropathy. It is difficult to say whether it is worsening due to eribulin or not, or it is persistent due to previous toxicity. But if quality of life we talk about eribulin versus taxanes, then it is better with eribulin than the taxane, because maximum patient out of 10 or 3 or 4 usually comes with myalgia always in every cycle of regular taxane.
Right.
Yes,
So,
the quality of life is definitely different, and particularly one another oral capecitabine also. It has been seen that as far as the quality of life is concerned, eribulin is much better tolerated than oral capecitabine.
so I think, yes, I generally agree. Anybody else? Dr. Vinu, on quality of life or neuropathy, please?
Yes, sir. Likewise, I think with eribulin not seen a deterioration of neuropathy. The pre-existing neuropathy we're able to handle with that, and definitely quality of life is much better. Like sir said, even better than capecitabine, I would say, where we sometimes struggle to even reach the full dose.
Right. So basically, by and large, well tolerated. Neutropenia would be a concern, which of course can be taken care of by the use of GCSF. So I think alopecia remains one issue which does happen, and fatigue, easy fatigability is another issue. But I think if somebody is responding, if the disease burden is going down, overall quality of life, the tolerance issues are so much better, especially than as compared to drugs like anthracyclines. Next question, if there's any. Okay, what line of treatment for eribulin in hormone receptor positive MBC patients? So I think Dr. Ankit did show, a slide sequencing the drugs. So there are, a plethora of drugs today, and you are guided by so many factors because you have HER2 low, you have BRCA1, 2, and of course you have PIK3C and so on. So Satyajit, what do you think? Where do you place eribulin?
Sir, in hormone positive patients, MBC patients, after third line, because of after exhausting all endocrine therapy. If it is endocrine refractory, then
Right.
it will
So can we just, count first line is going to be most likely AI with the CDK4/6?
CDK4/6 with AI. Then we will look for any resistant mutation like PIK3 or else. If it is positive, then we use that PIK3 inhibitor, everolimus, exemestane. Then later on, if in visceral crisis early has, then we can use in the third line also. Otherwise, beyond third line.
Right. So I think there's the issue of HER2 low. If somebody can afford, and failed 2 lines of treatment today, Destiny 06 allows using TDXD also. So taxanes are also there. So, if somebody is BRCA1, 2, then you have the PARP inhibitors. So I think eribulin is coming up definitely in third, fourth line, depending upon the number of hormones, PIK3C, HER2 low, PARP inhibitors, and so on. Anybody else wants to comment on the future role of eribulin in hormone receptor positive patient? Dr. Nikhil, any comments, please?
Yes, sir. Basically, as Satyajit mentioned, till now we will preferably use hormone treatment only for metastatic breast cancer who are not in visceral crisis. And for second or third line, I would like to use eribulin, especially in those with high Ki67. usually high Ki67, luminal B, or low hormone positive patients are usually refractory. In such patient, my preference will be the eribulin, definitely, because there is a data of
Like to use ahead of taxanes? That's my question.
definitely. Because nowadays when data is there, the eribulin early use will show conversion, hormonal conversion. It convert luminal B into luminal A. That is appearing a new data. So I would like to use in third line. Means after hormonal finish of hormonal, I'll go for eribulin, definitely.
Nirmal, you agree using ahead of taxanes?
Given the Destiny 06 data, I think really hormone positive and HER2 negative will be 85% will be HER low or ultra low. So they
Correct.
would qualify for TDXD. So I think eribulin, the role is going down and down in hormone positive especially.
Basically, TDXD, I agree, but the problem is the cost. Is I think even is
I can see Dr. Mahati. Is she there? She's not commenting.
Yes, sir. I'm here.
So any comments from your side on the use of eribulin or any experience you want to share for eribulin, please?
So in hormone receptor positive patients, I agree with Dr. Nirmal that eribulin is probably down the line, not even third, maybe down the line.
Right. So any experience sharing on triple negative or otherwise? Any long-term survival with eribulin?
Triple negative, we have got good outcomes in my clinical practice, sir. Usually I go with either second or third line. And I've already used eribulin with trastuzumab as combination in HER2 positive also. We
Right.
had a couple of patients who have progressed on HER2 and TDM1. So as fourth line, and they were not affordable for TDX. So went ahead with eribulin plus trastuzumab. They had a PFF for about 5 to 6 months, both the patients. So I thought that was a good PFS for fourth line. Like.
Yeah, I think you brought up the issue of HER2-neu, which is very, very important because the Emerald study, compared taxane plus trastuzumab and pertuzumab with eribulin along with dual HER2 targeting, and actually it was found to be equivalent in the Emerald study. So if somebody is not able to tolerate or has some kind of hypersensitivity issues to taxane. So I think eribulin can be substituted in first line for patients with HER2 positive MBC in combination with the trastuzumab, pertuzumab. And any other thoughts on use of eribulin in any other situation like sarcomas and other indications? Because that's another area where eribulin has been approved. That is the liposarcoma and leiomyosarcoma, particularly after anthracycline failure.
In sarcomas post anthracycline, there's a role for eribulin, sir.
Yes, absolutely.
The second or third line.
It is routinely being prescribed. Dr. Mukesh, any thoughts?
Sarcomas, obviously, post sarcomas we do use in third line or beyond, and it is a good molecule with lesser toxicity and have a response rates of maybe 40 to 50%.
Great. So, is there another slide? Okay, I think we already covered this issue. I only said about the the use of eribulin in Aditi is there, Dr. Aditi? Yeah, so I think we already covered this because eribulin can be combined with Trastuzumab and pertuzumab in the first line if you're not happy with the taxane somehow. And the results from the Japanese study, the Emerald 3 study, I think were equivalent in terms of PFS. So, because in patients who are on taxane with trastuzumab, who can't afford pertuzumab and maybe those other expensive molecules, thought could be to change the chemo partner, continue with trastuzumab, and substitute taxane with eribulin. So I think it is not a bad idea there. Satyajit, any thoughts on HER2 positive?
Sir, after 2 lines, third line we can use, and we can combine with trastuzumab first. Not sure with pertuzumab, but yes, can with trastuzumab.
I think you do have several drugs such as TDM1, and of course you do have Enhertu today. So if somebody can afford those drugs, so eribulin perhaps will go down the line, unless you want to just continue with trastuzumab and change the chemo partner, as I said. So is there another slide, please? The efficacy of eribulin in TNBC and non-TNBC patient. And again, Dr. Mahathi, your thoughts?
Yes, sir. Thing is, like, we tend to use Teribulin more in TNBC and in earlier setting in TNBC rather than non-TNBC patients. So that might confound the results, especially in HER2. In hormone positive, use it quite down the line as 4th or 5th. So
Right.
I would be using it more in TNBC in earlier settings.
Yeah, so I think
it's more efficacious.
So I think the forest plot which we saw in the meta-analysis did show that patients with visceral disease, non-visceral disease, TNBC or non-TNBC, they tend to benefit. But yes, TNBC tend to benefit more than the HR positive patients. HER2, I think we get a virtually equivalent benefit when we combine with the dual HER2 combination. But I think the hormonal therapy by and large takes the cake, and eribulin kind of comes a little down the line in those patients. So I think we are beyond our time. Next slide, please. Yeah, interesting question. Average duration of treatment of eribulin in your clinical practice. Do we have Dr. Durai Manavalan, please? Yeah, I didn't see him in the, the participants list. Anybody wants to take this? Dr. Nikhil?
Yes, sir. So those patients who are responding to eribulin on an average around 6 to 8 months is the median response rate I have seen.
Yeah, so I think that's usually the duration of response, around 6 months.
Around 6
Dr. Mukesh? Yeah.
Around same, sir, around 5 to 6 months, sir, mostly.
Sometimes 6 to 8 months, I would say, would be the average duration of treatment. I mean, we had a patient who went on for almost 1.5 years on eribulin. So the tolerance was good, the response was good, but alopecia remains an issue in this. And neutropenia, of course, as you keep doing the chemotherapy. Next slide. Yeah, okay. So I think we've covered most of the points in terms of side effects, efficacy, various subtypes of breast cancer. So I think we have this new drug, not new, we've been using it now for almost 4 years or 5 years, and easy to give, and quality of life is well maintained and is effective in all subtypes of breast cancer, particularly in TNBC and in HER2 positive. So I think that's another important area which should be sort of considered. So with this, I thank you all for this interactive session on eribulin and breast cancer. And thanks to Genova for giving us this opportunity to come together. Thank you.
Thank you.
Thank you so much, sir, for this wonderful delegation. And thank you all the experts as well for giving your clinical opinion on eribulin, especially in the MBC setting. And I think all of us in the audience have gained a lot of insights from the clinical pearls that you have shared. So thank you so much, Dr. Jain again, Dr. Shyam Agarwal, and all the experts for coming together today evening and discussing regarding this important topic. So thank you so much, everyone, for joining in. See you for the next one. Good night.